Hello everyone and welcome to today's webinar entitled Bio-Inspired Materials Using Genomics to Engineer Recyclable Materials. My name is Dawn Van Dam and I'm the General Manager of Cambridge Health Tech Associates, and we are the organizers of the Next Generation Sequencing Leaders online community. So I'll just do a brief introduction for today's speaker. So this is a webinar using genomics, talking about using genomics to engineer recyclable materials. Dr. Melik Demirel, Professor of Engineering, Materials Research Institute, and Huck Institutes of Life Sciences at Pennsylvania State University, will be discussing the use of elastomers to expedite the process of creating eco-friendly materials that can be manufactured into nano and micro-scale devices. Bio-derived or natural materials have been a fundamental part of human life since the dawn of civilization. However, due to exploitation of natural resources and cost issues, synthetic materials are replacing bio-derived materials in many industrial products. There is, accordingly, an ongoing and unmet need to develop novel materials that can be produced easily from renewable resources at reduced cost and in a broad array of useful applications. Using next-generation sequencing in conjunction with high-throughput proteomics and advanced materials characterization, it is now possible to rapidly characterize useful protein-based model systems and to later express them by recombinant expression for downstream development, according to an approach developed by Guerette, Hoon, and co-workers. Alternatively, direct extraction of elastomers from biological organisms also provides a new generation of recyclable-engineered materials. Understanding the structures and functional characteristics of these novel elastomeric materials will expedite the design, fabrication, and synthesis of eco-friendly, recyclable, advanced materials with resilience to environmental factors and novel wetting, friction, and transport properties. The original paper describing the biometrics research in the context of transport, adhesion, and wetting was published in Advanced Functional Materials according to the bibliographic references listed on our website. So I'm going to move to the next slide, or ask to have the next slide be moved to, and we'll tell you a little bit about our organization, and then I'll introduce Dr. Melik Demirel for today's presentation. So Cambridge Health Tech Associates is an organization that specializes in providing a number of services to the marketplace. That would be including our online community called Next Generation Sequencing Leaders. And through Next Generation Sequencing Leaders, we do discussion forums, blogs, networking, webinars, focus groups, product reviews, resource library, and scientific posters. So I'm just going to make sure everyone is on slide number 2 here. NGS Leaders, as I mentioned before, is organized and managed by Cambridge Health Tech Associates. And what we are really contributing to the science of life sciences and healthcare is helping transform that science, or your science, into commercial success. So we're proud to be able to offer a presentation today to show how this can move forward in the marketplace. In order to help you do that, transform your science into commercial success, we have 3 different services that we offer to the marketplace. And then the first is consulting and market research. And here we can help you with an analysis of the marketplace, understanding what your customers need and want. The second thing that we do is we have a technology evaluation consortium where we bring together new technologies and we share them with the pharmaceutical and biotech industry and get them to evaluate them. And the third thing that we offer is marketing and communication services, as well as our online communities, in order to help spread the word and communicate the great science and thought leadership that's going on out there in the industry. We did launch a new division of our organization about a year or so ago, and we still are working quite diligently with Dr. Kevin Davis, who's a specialist in next-generation sequencing and genomics. And if you're interested in working with him and us in looking at any of your market research or business planning needs, we would be more than happy to do that for you. So with no further ado, I would like to introduce our speaker for today. So Dr. Melik Demirel joined Pennsylvania State University as the Pierce Development Tenure Track Assistant Professor in the fall of 2003, following completion of his PhD degree in Materials Science and Engineering at Carnegie Mellon University and an appointment as a postdoctoral scientist in the Theoretical Division at the Los Alamos National Laboratory. He was also hired as a group faculty to pioneer the new area of bio-nanotechnology at Penn State. He was promoted to the rank of Associate Professor in 2008 and Full Professor in 2013. His achievements, in part, have been recognized through the receipt of a Young Investigator Award from the Department of Defense, an Alexander von Humboldt Fellowship at Max Planck Institute for Biophysical Chemistry, the Wyss Institute Visiting Scholar at Harvard University, an Institute for Complex Adaptive Matter Junior Fellowship, the Nicholas and Gelsa Pellick Biotechnology Innovation Award, and the Pierce Development Professorship at Penn State. He has been frequently invited as a lecturer to multiple national and international congresses, workshops, and courses, and his research focuses on theory-driven functional material synthesis and fabrication to produce next-generation materials for an array of fields including energy, biomedicine, and security defense. So with no further ado, I will pass the baton over to Dr. Melik Demirel and let him take you through his presentation for today.
Thank you, Dawn. Hello, everyone. Thank you for inviting me to present an online talk on nanoscale biomimetics. I would like to thank to the attendees, and please feel free to contact me if you have any question at the end of the talk.
Oh, sorry, I just forgot to say one thing, Dr. Demirel, is if anyone has any questions as you go along, if you could please just put the questions in the chat box, and then at the end of the presentation, I will ask the questions of Dr. Demirel, and he will answer them. And the second thing is that we will also be posting an online survey at the end of the presentation to get your feedback on the presentation and look for future ideas from you and comments. So again, I'll pass it over to you, Dr. Demirel.
Thank you. Three aspects of creating eco-friendly materials include discovery, synthesis, and biomimicry. Combining these three approaches allows rapid characterization of natural materials and the translation of their molecular design into usable, unique materials. Our work began with investigating biological asymmetric structures, trying to understand how nature solves problems, such as butterfly wings' ability to shed water and gecko feet's capacity for scaling walls. These concepts were then used to mimic natural structures to create tools and devices on the micro and nano scales. Recently, integrating these concepts into eco-friendly devices for wetting, friction, and transfers constitute an important part of our research. Together with my collaborators around the world, we extended these studies to materials extracted from biological sources. As the materials science expert, we further developed functional materials that can be produced easily from renewable sources, which would offer a broad array of useful applications. The materials can be shaped into almost anything: nanotubes, fibers, gels, thin films, and colloids. Specifically, an integrative approach for the understanding and engineering of elastomeric materials has been developed. This approach opens up a new field of functional biomimetic materials for manufacturing eco-friendly advanced materials and devices. Let me introduce the biomimetics for you. The biomimetics is the adaptation of methods and systems found in nature into synthetic construct and microscale devices. Microscale biomimetics are probably the most developed. They commonly involve systems mimicking structures or physical properties of a natural system. On the other hand, nanoscale biomimetics are based on assembly of molecules or macromolecules complexes. A variety of devices have been realized at this level at the microscale, ranging from nanoscale sensors for detection of analytes to prototype devices capable of novel wetting, adhesion, and transport properties that I will discuss today. Natural materials have been a fundamental part of human life since the dawn of civilization. The Silk Road, for example, extends over 4,000 miles that get its name from the Chinese Silk Trade began during the Han Dynasty around 2000 BC. In this slide, you see the profiles of 6 natural major plant and animal fibers and their electron microscope images. Plant fibers include seed hairs such as cotton, stem fibers such as flax, leaf fibers such as shisha. Animal fibers include wool, hair, and silk. Natural fibers are used in many industrial products as the environmentally friendly choice. For example, European car industry is currently replacing glass fibers in plastic car panels with flax fibers, which reduces skin and respiratory irritation. Additionally, low weight, which cuts the fuel consumption of natural fibers, made it attractive to the industrial applications. In the textile industry, the biocompatibility of natural fibers makes their wearers less prone to skin rashes, itching, and allergies, often caused by synthetic formats. The cost and pollution associated with the transportation of this material is minimal because many of these materials are available throughout the world. Other examples of innovation are in the area of structural engineering, where cement blocks reinforced with fibers are now being manufactured in Tanzania and Brazil. There is already a sizable market for natural fibers of $36 billion a year, according to a report published in 2008. Note that 10% of the market is the protein-based natural fibers such as silk and wool. Bio-derived materials are studied extensively in the literature. There are over 100 books and more than 100,000 citations in the literature. A Google Scholar shows an exponential increase in the citation for silk, elastin, and resin, the most 3 common bioelastomers in the last decade. Functional biomaterials made of natural building blocks include protein, polysaccharides, lipids, or nucleic acids. There are 68 known building blocks, 8 of which are nucleotides, 32 are glycans, 20 are amino acids, and 8 from lipids. Because of their advantage, including well-defined structural features, unique shapes and sizes, genetic programmability, and robust chemistries, these macromolecules offer great opportunity for functional material fabrication. Proteins are linear polymers that each can contain only one basic type of linkage between monomers, such as amino acids. In contrast, each monosaccharide can generate theoretically any of several positions of another monosaccharide in a chain. Thus, it has been pointed out that although 3 different amino acids can only generate 6 trimers, 3 different glycans could produce over 1,000 unique 3 saccharides. Thus, an almost unimaginable number of possible saccharide units could be theoretically presented in biological systems. However, biological polysaccharides cannot be coded because they have no genomic information and they are generated through metabolic pathways. Compared to proteins, such as gene products, can have functionalities and conjugation chemistries with limited amounts. A direct comparison of mechanical properties of natural and engineering materials reveal that nature is able to synthesize materials with properties that cover a range almost as large as those in engineering materials we use today. However, cost is a major competitive disadvantage for natural materials. The total global consumption of polymers in solid forms was 160 million tons in 2003 and 2020 million currently. Bioplastics represents only 0.1% of this amount in 2007. This is due to the lack of natural resources or the cost issues. While the principal raw materials for the production of organic materials during 1930s were coal and carbohydrates and petroleum, by 1960s, petroleum and natural gas supplied more than 80% of all industrial organic chemicals. Therefore, in the last century, we have been heavily relying on petroleum for the production of plastics and textiles. Processing and recycling of synthetic materials leads to high level of water pollutants and persistent chemicals, including heavy metals released in the processing. In contrast, bio-derived materials really excel in disposal stage of their life cycle. They decay through the action of fungi and bacteria with minimal cost to the environment. There is accordingly an ongoing and unmet need to develop novel materials that can be produced easily from renewable resources at reduced cost and in a broad array of useful applications. Such demands cannot be met without fundamental breakthroughs in the area of nanoscience and engineering. The most recent attempts to mimic biopolymer function is the synthesis of non-natural folding polymers such as foldamers. Foldamers adopt complex folding topologies via sequence-defined structures similar to proteins. Although they are proposed to drastically improve our capabilities in enhanced stability, new folding topologies, and tailored functions, their various synthesized foldamers are limited because there is no available approach for rapidly developing and screening large-scale non-natural polymer libraries. According to the environmental conditions, proteins can be roughly divided into three classes. Fibrous proteins form vast, usually water-deficient aggregates. Their structure is usually highly hydrogen bonded, highly regular, and maintains mainly interactions between various chains. Many bioelastomers have been extensively studied, such as elastin, collagen, silk, keratin, and resin. In general, these families of structural proteins are characterized by long-range order molecular secondary structures, such as beta sheets, coil interactions, and triple helices. These features promote self-assembly, the formation of structural hierarchy, and thus materials related to functions in nature. The other category is the membrane proteins, which reside in a water-deficient membrane environment, and their intramembrane portions are highly regular but restricted in size by the membrane thickness. And the last category, which is the most common one, is the globular proteins, which are less regular, and their structure is maintained by interactions of chains with itself through hydrophobic or hydrophilic interactions. Although the current elastomers, bioelastomers, are limited to medical markets, such as tissue engineering, bioadhesives, and etc., several companies have set up laboratories and research teams to find uses of bioelastomers in high-yield and low-cost industrial products. Nanotexture surfaces composed of asymmetric structures are widely used in nature for wet and dry adhesion. Electron microscope images shown in the slides of various structural features. Some insects employ a special form of wet adhesion by secreting adhesive fluids through arrays of setae to adhere to smooth or rough substrates. For example, the beetle uses such a technique to respond to disturbance by activating a tarsal adhesion mechanism. While defending itself from a predator, the beetle presses a surface with all its bristles to withstand pulling forces of up to 60 times of its body mass for up to 2 minutes. The flexible hairs on geckos allow a much greater contact area with uneven surfaces compared to smooth surfaces. The density of hairs on such surfaces scales with the body mass. Insects, for example, have lower hair densities compared to lizards. The force of single seta of gecko toe pads, measured by using a two-dimensional micromechanical system, shows that the attachment forces are 600 times greater compared to the flat surfaces. Water repellency is a critical feature of water walkers for avoiding entrapment at the surface, surviving the impact of raindrops and submerged breathing. Butterflies, for example, have superhydrophobic wings composed of overlapping strips with directed flexible nanotips. Nanotexture surfaces are also common in plant world. Pitcher plants have directional leaves that capture, retain, and digest insects. Understanding the structure and functional characteristics of elastomeric materials that are shown in the previous slide will expedite the design, fabrication, and synthesis of eco-friendly materials. The array of techniques for fabricating nanoscale biomimetic surfaces can be categorized as rigid structures, stimuli responsive structures, and adaptive surfaces. Numerous techniques for manufacturing rigid surfaces for rigid anisotropic surfaces have been developed, ranging from template and template-free deposition to photolithography to direct printing. Here, I will discuss the application of nanoscale biomimetics. The first example is from Dr. Metin C's group at Carnegie Mellon University in collaboration with Dr. Rogers' group at University of Illinois. Gecko-inspired angle elastomer micropillars are fabricated as compliant pick-and-place micro-manipulators. By gently pressing the tip of a pillar to a part, the pillar adheres to it through intermolecular forces. Next, by retracting quickly, the part is picked from a substrate. They show that during transferring, the adhesion between the pillar and the part is high enough to withstand disturbances due to external forces or weight of the part. During release of the part onto a receiver substrate, the contact area of the pillar to the part is drastically reduced by controlled vertical or shear displacement, which results in reduced adhesive forces. They also claim that the compliant polymer micropillars are safe to use with fragile parts and could be effective on most materials and in air, vacuum, and liquid environments. This work was published in Advanced Materials recently. The second example is from my group. We reported an engineered nanofilm composed of an array of nanorods which demonstrate asymmetric wetting behavior by means of a pin-release droplet ratchet mechanism. The nanofilm provides a microscale smooth roughness, microscale smooth surface on which it transports microliter droplets, and it is also relatively easy to synthesize by a bottom-up vapor phase deposition technique. This work was published in Nature Materials in 2010. We also demonstrated the actuation of microgel encapsulated in droplets using these surfaces, which offer a pathway towards high throughput microgel assembly applications to build complex constructs. The final example is from Dr. Taksin Wong's group at Penn State in collaboration with Dr. Joanna Eisenberg group at Harvard. They have created liquid repellent microtexture surfaces inspired from pitcher plant surfaces. Other non-wetting surfaces that rely on the formation of a stable air-liquid interface have been developed, but they were limited in oleophobicity, which failure under pressure and upon physical damage, inability to self-heal, and high production costs were listed. To address these challenges, they reported a strategy to create self-healing, slippery, liquid-infused porous surfaces with exceptional liquid and ice repellency, pressure stability, and enhanced optical transparency have been achieved. They show that these properties are insensitive to precise geometry of underlying substrates, making their approach applicable to various inexpensive, low-surface energy structure materials. They also claim that these surfaces will be useful in fluid handling and transportation, optical sensing, medicine, as well as cleaning and anti-fouling materials operating in extreme environments. Although tremendous progress has been achieved in these areas, there are still limitations in the fabrication of biomimetic materials. These are lack of reversible, renewable materials in material fabrication and limited assembly of these materials into 3D constructs, which I will mention in my upcoming slides. Recombinant expression or direct extraction of elastomers from biological organisms can provide a new generation of recyclable engineering materials. Although recombinant expression of bioelastomers has been achieved in the last 2 decades, for example, silk and elastin, I will focus on the squid ring thin, which is an ideal recyclable engineering material due to its reversible aggregation mechanism. And it also addresses some of the limitations that I mentioned in the previous slide. This property is the key for biomimetic manufacturing at the nanoscale, which is the main focus of this talk. There is an ongoing and unmet need to develop novel materials that can be produced easily from renewable resources and which would offer a broad array of usable applications. Efforts to engineer new materials inspired by biological structures are hampered by the lack of genomic data from model organisms studied in biomimetic research. Recently, Dr. Ali Mizares at Singapore and his collaborators showed that integrating high-throughput RNA sequencing with proteomics and advanced material characterization accelerates biomimetic engineering. This approach can be applied to a broad range of systems such as egg case proteins, adhesive proteins in green mussels, and squid sucker ring teeth. I will focus on the squid ring teeth only because of its reversible aggregation mechanism, which is extremely important for nanoscale biomimetic manufacturing. About a book published by Williams a century ago in 1910, entitled Anatomy of Squids, studied the squid arms and tentacles in detail. At the time, the author claimed that the squid teeth is composed of chitin-like substance, but it differs from chitin since the new material is soluble in dilute alkaline solutions. The tissue surrounding the squid's ring tip is well described in the book. The inner surface of the side of the cup is covered by a columnar epithelial which secretes the ring and whose height at each point is proportional to the thickness of the corresponding portion of the ring. The position of the nuclei of this cell varies greatly. In some cells, the nuclei are at the base, in other in the middle, and yet other at the free end of the cell. 4 decades ago, Nixon and Dilley studied the suckers in cephalopods in an article published in 1977, and they concluded that squid suckers have teeth that are made of proteins but not chitin-like. They also show that SRT structure has pores. Recently, the amino acid composition and mechanical properties of SRT are studied and compared to other bioelastomers by Dr. Mizares' group and his collaborators. Last year, in collaboration with Dr. Mizares' group, he, for the first time, demonstrated the fabrication of nanoscale structures using SRT. This work was published with Dr. Mizares' group, which I will describe in the next slide. Next-generation sequencing approaches provide genomic studies for a wide range of biological elastomeric materials in species whose genomes have not been sequenced. Here, I'll describe an approach which is slightly different than the approach that is introduced by Dr. Mizares' group for characterizing bioelastomers. The bioinformatic analysis that they have performed can be thought as an iterative, superimposed process that uses information from several experiment measurements and combines multiple bioinformatic techniques. We started with a transcriptome assembly with the Trinity software. The reference for the Trinity software is given below. This process produced a large number of transcripts that represent the entire cellular mRNA content of the cells that form the suction cups of the SRT. We then used a second source of information, that of the peptide sequence produced via mass spectroscopy instruments, which will be described in the next slide. This allowed us to identify those Trinity-produced transcripts that were likely to match the protein fragments of interest. Finally, we made use of a third source of information, that of the presence of the beta sheet with a known sequence pattern, and filtered out transcripts to keep those that match with this pattern. We call the transcripts that match all 3 sources of information as the candidate transcripts, and we consider this to be the the fragments of the proteins of interest. Peptide sequencing from the whole SRC protein complex were sequenced using mass spectroscopy and matched against the Trinity assembled transcripts shown in the previous slide. De novo sequencing identified peptides directly from tandem mass spectra. More than 15 de novo sequencing algorithms have been developed in the last 2 decades, and 5 of the most common algorithms that are compared in detail in the article cited below of the slide. Following these steps of mass spectroscopy analysis, race PCR and sequencing steps are implemented to verify the protein of interest. I should emphasize that these last 2 steps are very important because the Trinity software that I described in the previous slide could fail because of an internal repeating region in the genomics sequences, which is common in bioelastomers. Thermoplastic proteins have been known in food industry for a long time. Solution casting is the most commonly used method to form protein-based films in laboratories. However, that method is an expensive process and has some limitations when it comes to the fast and continuous industrial processes of plastic films. Thermoplastic processes used in plastic industry are considered to be preferred because drying steps could be eliminated, which accelerates the processing times. Utilization of extrusion and injection molding techniques has been the advantage of offering low-cost and versatile production systems. Plasticizers are generally added to the protein matrix to improve processability and to modify the protein's properties of the final structure. Common plasticizers are listed in the table, and they are summarized in a recent review cited below. Water is the most effective plasticizer in biopolymer materials, enabling them to undergo the glass transition, facilitating deformation and processability of the biopolymer matrix. Without water addition, the temperature region of the thermal degradation would be easily reached before film could be formed. For example, for the squid ring thing, the degradation temperature is around 196 Celsius. However, an excessive amount of water during the protein extrusion would decrease the melt viscosity. Besides water, glycerol is a low molecular weight hydrophilic plasticizer that has been widely used in thermoplastic processing of proteins. Its high plasticizing effect has been attributed to the ease with which glycerol can insert and position itself within the 3-dimensional protein structure. The protein melt is molded into thin ribbons, as shown in the figure, to perform mechanical analysis to discover the elastic modulus of the squid ring teeth. The stiffness values of 1 gigapascal in dry and underwater conditions at room temperature are measured. The modulus of the elastomer drops to 10 megapascal above the glass transition, which is similar to the modulus of elastin. Fabrication and synthesizing materials resilient to environmental factors have been achieved by studying functional transition of biomimetic systems both computationally and experimentally. The long-term engineering impacts of this research includes new design rules for protein assembly to engineer novel structures for manufacturing nanoscale products and devices, novel micro and nanoscale tools and devices for researchers in the biotechnology and medical field, and production of low-cost bio-derived elastomers that are superior to alternatives to synthetic plastics that minimize waste and pollution. Future areas of research for bioelastomers include renewable carbon fibers for transport industry and biologically derived fibers as an alternative to fibers such as nylon for textile industry. In summary, I show that the discovery and recombinant expression of bioelastomers open up new avenues for materials fabrication and synthesis, which will eventually be competitive with the synthetic oil-based plastics. Recombinant expression or direct extraction of elastomers from biological organisms can provide a new generation of recyclable engineering materials. Bioelastomers are reusable, recyclable, and biocompatible, and provide multifunctional properties. Elastomers are, as I mentioned, ubiquitous in nature. In plant and animal kingdoms, directional surfaces with microscale and nanoscale features provide key functions for survival, such as transport, adhesion, and wetting. Mimicking these structures will open up a new avenue for nanoscale devices. I would like to also suggest the following books and articles for further reading if you are interested in this area. The book that's published by Stanford and Reynolds, Nature's Robot, describes the concise history of natural fibers, and the book that's published by Finkelstein and Stilson describes the protein physics. And another book on capillary and wetting phenomena describes the physical view of adhesion and wetting. Some of the most important RNA sequence and mass spec articles are listed as well. And a review article describing the biomimetic issues in advanced nanoscale biomimetics is published in Advanced Functional Materials recently by my group. And also a good review to read about protein-based composite materials is published by David Kaplan's group at Tufts University. Finally, we would like to acknowledge the support for this research from Office of Naval Research and Pennsylvania State University. I would like to thank also to my past and current group members and collaborators around the world at Penn State, and now I'm happy to answer any questions.
Great. Well, thank you very much, Dr. Melik Demirel. We really appreciate you taking the time today to share this review with us and to share the science that's happening in this area. So again, I'll just remind everyone to put any questions into the chat box, and just above that is also the survey that we would love you to fill out. So I'm going to jump into a couple questions that have come in already, and I would, as I said, invite people to put in additional questions that you'd like to have answered. So Dr. Demirel, you provided various examples of structural nanoscale features from the animal and plant kingdom. What are the limitations of fabrication or design of similar structures in the lab?
So the limitations specifically are in the area of renewable materials, such as the one that I described today. The Squid ring tint is a good renewable materials that could address this area of fabrication. And the other area is the limited assembly of these structures into three-dimensional constructs to fabricate three-dimensional materials.
Okay, great. Thank you very much. So one question here is, why do you study squid ring teeth or SRT protein? What is the motivation for working with SRT?
So SRT has a unique ability, which is basically a recyclable engineering material due to its reversible aggregation mechanism. Unlike other bioelastomers, this mechanism provides two phases. One is in solution and other is in the elastomeric phase. Therefore, this material could provide a key advantage compared to other bioelastomers for manufacturing at the nanoscale.
Okay, great, thank you. So recombinant expression of bioelastomers, for example, silk and elastin, has been shown a decade ago. So what is new about SRT or squid ring teeth protein?
Efforts to engineer new materials inspired by biological structures are hampered by the lack of genomic data from many model organisms studied in biomimetic research. Together with our collaborators, recently it has been shown that the high throughput RNA sequences with proteomics approaches can accelerate this biomimetic engineering. This approach can be applied to a broad range of systems, as I illustrated in the presentation today.
Okay. So here's another question, and again, I invite people to continue asking more questions. What is nanoscale biomimetics, and how does it differ from conventional biomimicry?
Okay, so biomimetics is the use of and the implementation of concepts and principles from nature to design functional materials or devices or systems. This adaptation of methods and systems found in nature into synthetic constructs is desirable because evolutionary constraints forces natural systems to become highly efficient. Microscale biomimetics are probably the most developed one, but nanoscale biomimetics is recently being developed by several groups around the world. A variety of devices by nanoscale biomimetics I already have described, the micromanipulation surfaces that can create oleophobic and oleophobic surfaces, and other nanoscale surfaces which can create directional transport.
Okay, great, thank you. Thank you for that answer. That was terrific. So another question for you: what are the challenges in compounding the SRT into adhesives using traditional plasticizers?
Okay, so the high viscosity is a big challenge of SRT to be used in intrusion and extrusion processes. And other properties, other challenges include the temperature dependence, the TG glass transition dependence on the plasticizer that is used.
Okay. Another question is, are there any bioelastomers produced currently for commercial purposes using recombination?
Yes, the silk and elastin has been recombinantly expressed over 2 decades, and there are companies which are selling this product, which are available for purchase.
Okay. Okay, and another question would be I think it's how are we able to produce bio nanomaterials that can be price competitive?
That's a very good question. This is one of the biggest challenges today. So the production cost of recombinant expressed protein is in the order of $50 to $60 per gram. Okay, so this is a huge number compared to synthetic materials that are produced at $1 per kilogram. So, but there are alternative techniques to reduce the cost by using high-scale production in different plants or organisms, so that will open up the new low-cost materials in the near future.
Okay, great, thank you. Are there known plant bioelastomers?
Yes, so several bioelastomers that I mentioned in my talk, soybean protein, wheat gluten, and so on. That these are the thermoplastic proteins that I mentioned in my talk.
Okay, and are there limitations to scale up quantities?
The current limitations to scale up quantities are limited by the fermentation facilities, that are common techniques that use, bacterial expression of the proteins. So the yield is one limitation, and the cost is the other limitation in this area.
Okay, great, thank you. What are some of the unique advantages of SRT bioelastomer compared to other elastomers, for example, resin, abductin, silk, etc.?
So as I mentioned, the SRT has a very interesting reversible aggregation mechanism, and it has a glass transition at body temperature in water, which in addition has a very strong elastic modulus in both wet and dry conditions of 1 gigapascal. So these are a unique advantage compared to the other elastomers.
Okay, great, thank you. So could you talk about the durability of these nanostructures from SRTs? Many times synthetic nanostructures have poor durability and therefore the properties are not sustainable. Are natural nanostructures more durable compared to synthetic nanostructures?
Well, this question has to be answered in the context of environment and conditions, because depending on the environmental conditions, the durability may change. But it is well known that these proteins that are extracted from biological sources are stable to many enzymes and environmental conditions, but they are, for example, SRT could be soluble in. It's known that over a century that it could be soluble in alkaline solutions such as acetic acid, formic acid, and so on. So the answer to this question depends on environmental conditions and chemicals that are used in the durability processes.
Okay, great, thank you. Another question here is, have you tried to direct cell migration similarly with a droplet?
Yes, cell migration has been studied not by our group, but other groups have shown that ratchet surfaces can direct cell migration. What we have done in collaboration with Professor Christian Steyer from Tufts University is directing neural growth, the axon growth of neurons, which we published in the Applied Physics Journal a couple of years ago, and another paper has been recently submitted, which we have shown that we can control the axon growth of the neurons unidirectionally, and we can study their dynamics in real time.
Okay, great. Well, thank you. Another question for you here is, can you comment on natural fiber properties obtained from polysaccharides, for example, cellulose, versus proteins?
Okay. So proteins are linear polymers that can contain only one basic type of repeating unit, such as amino acids, as I mentioned, right? So in contrast, monosaccharides, for example, which is made of the cellulose, can theoretically generate linkage to any of several positions of another monosaccharide in a chain. Thus, it has been pointed out that 3 different, for example, amino acids can generate only 3 trimers, but the glycans could generate over 1,000 unique structures. So this is the major advantage of cellulose compared to proteins. However, the disadvantage is the polysaccharides cannot be coded. So because there is no genetic information and it's the end product of metabolic pathways, so the limitation of the functional chemistries and conjugate chemistries of polysaccharides are the key limitations compared to the proteins.
Okay, thank you very much. So I do have a couple more questions, but I just want to remind everyone that if they have any more questions, to please put them in the chat box. So the next question here is, how about non-natural polymers? For example, foldamer that mimic the ability of macromolecules to fold into well-defined conformations compared to elastomers.
Non-natural polymers that mimic biological functions are generally called polymers. So although polymers can have enhanced stability and new folding topologies and expanded applications and tailored functions, their limitation has been the synthesis, which is to create a large scale non-natural polymer libraries to screen different properties have been the limitation compared to natural polymers, such as proteins, DNA, and etc.
Okay, thank you very much for the answer to that question. Given the high cost of protein-based fibers compared to synthetic fibers, what are the industrial applications?
So as I mentioned earlier in a previous question regarding to the price, the current bioelastomers are limited because of the high price, they are limited into the medical and pharmaceutical markets. And but several companies have set up laboratories and research teams to use bioelastomers in high yield and low cost industrial products. And I believe in the next decade or so, we will have low cost and highly usable natural fibers in the market.
Okay, thanks very much. And so one last question, and just to remind everyone to fill out that survey while we're doing this last question. Can you speculate on future areas of research for bioelastomers?
Yes, there are multiple areas that bioelastomers can excel, and one of them is the renewable carbon fibers for transport industry that could be made from bioelastomers. And the other is in the textile industry, where biologically derived fibers could be used as an alternative to synthetic fibers such as nylon.
Okay, great. Well, again, just wanted to say thank you very much to Dr. Melik Demirel for his presentation today. And if anyone has any questions or would like to discuss this further, you can contact us or contact him directly, and we'll make sure he gets a message if you contact through us. So again, we appreciate everyone's attendance in today's webinar, and we look forward to future events like this to share the science and help transform everyone's science into commercial success. Thanks again, and have a great day.
Thank you.